Okay, so hello, from Life Education. My name is Harshita, and we're going to study the next lecture of a biological classification. And today's lecture, we are going to study monerans. So first of all, monerans were established by Copeland. Okay, so like we in the previous lecture we studied classification of like the classification system, how classification system evolved. So in evolution, we saw that first classification system was proposed by Linnaeus. There was two kingdom classification, in which he included plants and animals, only two things. After that, three kingdom classification came, which was established by Ernst Haeckel, right? In which three were there like plants, animals and protista. After that, four kingdom classification was there, which was proposed by Copeland, in which he included monerans for the first time. Okay, so right, we're going to study about monerans in more detail today. So basically, the basic structure of monera, the sole members of monera are bacteria. Like all the bacteria come under monerans, right? These bacteria are divided into two parts, which we are going to study further in more detail again. And these are archaebacteria and eubacteria. Archaebacteria and eubacteria. The division of bacteria is divided into archaebacteria and eubacteria. These monerans, these we can say these bacteria are ubiquitous. This is a line from NCERT. Now, what do we mean by ubiquitous? Right again. Okay, so by ubiquitous we means like cosmopolitan, we can say. Okay, another word for ubiquitous is cosmopolitan. I just made last way. Cosmopolitan. What is cosmopolitan or ubiquitous? It is means present everywhere. So ubiquitous or cosmopolitan means present everywhere, and we're talking about bacteria. So bacteria are ubiquitous, whereas they are present everywhere. Like we can take an example of lake, river, land, air, everywhere. Like we can say everything that we consider, like we human beings are confined to a place that is land, or we can say terrestrial. We are terrestrial animals, but these bacteria are cosmopolitan or ubiquitous. They are present everywhere. Even if we consider hot winds, like the thermal springs where the temperature is too high, like above 100 degrees Celsius, these bacteria can survive there also. Not all these bacteria, but some of these bacteria can. And marshy lands, okay, these are the example of archaebacteria. We're going to study further in detail, but I'm just giving you an overview of the chapter. So marshy areas, like the we can say कीचड़ वाले areas, basically the कीचड़ वाले areas where there is absence of oxygen, these bacteria can grow even there. Like they're anaerobic and obligatory anaerobic first, and they can survive in that place, and they can produce methane, that is biogas, from these methanogens. That is why they are known as methanogens. And one more example for archaebacteria is halophiles. Halophiles can live in salty areas, like extreme salty areas, in which we cannot survive, the fishes cannot survive, the bacteria can survive. So from this I mean like जितना भी हम लोगों ने यहाँ पे अभी तक देखा है उससे I just only want to say that bacteria are very important. They're like abundance is in millions. We can't count them. Like हम लोग जब भी we live in India, so जो हमारा पूरा वर्ल्ड है उसके लिए हमारी पॉपुलेशन काउंट की जाती है like every 5 years, like हमारी. So हम लोग पॉपुलेशन काउंट करते हैं. हम लोग there is also a process called DNA fingerprinting, in which the DNA of organisms are collected in databases and all these. This is also a hectic and lengthy process also. But for bacteria, we cannot even do that. cannot even do like computerically, like it's like impossible. These are in millions, and we do not even know some of these species also. So basically, what I was trying to tell you is, I just want you to feel what is bacteria, like what bacteria are. Okay, so let's do it. Okay, so now that we know what bacteria are, where bacteria are, and all these things, let's know about their structure. Like what make them survive in these extreme conditions, and also like we're not going to study what makes them survive in these conditions, because these are specifically confined for archaebacteria. We're not studying archaebacteria, but we are studying basic moneran characters, that is basic bacterial characters. So let's see what structure it is. Like we can study cell wall of bacteria. Cell wall of bacteria is made up of peptidoglycan, and this is an important point. But there is an exception to it, like all the bacteria's peptidoglycan is present, but exception. Except archaebacteria. Archaebacteria, and one more thing, mycoplasma. So let's see what makes them an exception. Archaebacteria are like a whole different kind of bacteria. Like I told you, they can survive in extreme conditions. So what makes them survive in extreme conditions is their, let's keep it a secret, right? Let's study the cell wall structure first. The cell wall is made up of glycoprotein, and we can say pseudomorine, so this is the constituent particles of the cell wall of archaebacteria. What makes mycoplasma the exception? We are so curious about that. Let's see. Mycoplasma are the bacteria, we can say these are the smallest bacteria, or even the smallest living organism. There is an exception that even in mycoplasma, the PPLO are the smallest. PPLO are like 0.01. Let's see. Wait a minute. Let's see it over here. Mycoplasma, smallest organism. Smallest organism in the whole world. So see, the size is 0.3 micrometer. So those who do not know what micrometer is, micro is 10 to the power minus 6 meter. So small, right? So small. Smallest organism, 0.3 micrometer. Bacteria are of course small. We can see them with our naked eyes, right? And even in these, PPLO are the smallest, 0.1 micrometer. So what PPLO are? These are pleuro, pneumonia like organisms. Pleuropneumonia like organism. PPLO are pleuropneumonia like organisms. So these are the smallest ones. Let's study. So we studied the cell structure, cell wall structure basically. Let's study further. Okay, so we are at this time that we have studied the cell structure. Let's see what shapes are bacteria present in. There are various shapes. We're going to study 4 shapes which are important because, okay, let me tell you one thing. From the last thing that we studied, like PPLO and mycoplasma, these are important things. The question can arise, what are the smallest organisms in the world? So if there is an option like there is only mycoplasma present, you're going to take mycoplasma. If there is PPLO and mycoplasma both are present, you're going to take PPLO. Okay, right here, right. Now let's study the shapes of bacteria. What shapes are bacteria? Like there are various shapes, but I'm gonna study four shapes. These four shapes are also like in detail if you're gonna study in detail. But in NCERT we only have these four shapes and the characteristic features of the shape. Like some examples of it. Let's see. Shapes of bacteria. There is Focus, Rhodo, Spirillum. Okay, let's see these two first. Focus is basically spherical. Rhodospirillum. Spirillum means spiral. Now next we have Vibrio. Vibrio is comma shape. And the last we have Bacillus. That basically is not last, but yeah. It is rod shape. So let's make the shapes out so that we can have feel of it. These are focus. It's not a sphere, yeah, quite similar to that. If we have to compare, no, Rhodospirillum, these are like this. These are like flagella on their both heads. This condition is known as amphitrachus if I'm not wrong, but yeah. These are spirillum. Next we have Vibrio. Vibrio are like comma shape. This is Vibrio. Bacillus, rod shaped bacteria. So this is what we can call shapes of bacteria. These are, now that we have studied the basic characteristics of monerans, the cell wall structure of monerans, the shapes of monerans, and let's see the mode of nutrition of monerans. Like these monerans are getting nutrition from somewhere that they are affecting us so much that there must have to be a nutrition mode of nutrition. Majority of the bacteria are heterotrophs. We can say clearly, like without shape. Majority are heterotrophs. And some are autotrophic. Which can be photoautotrophic or chemoautotrophic. Just complete the words. Okay, so mode of nutrition, majority of the bacteria are heterotrophs. So we're gonna we're gonna recap or revise what autotrophs and heterotrophs are. I know all of you know it, but still. Heterotrophs which gain the nutrition from other organisms. Like we are heterotrophs, the plants make their own food, we eat the food made by them. Understand? Like in heterotrophs वो लोग होते हैं, वो organisms होते हैं जो खुद खाना नहीं बनाते हैं, लोगों को खाना बनाना खाते हैं. We cannot say our mommy is an autotroph, no, she's not. Or the people who make food like chefs or anyone, they're not autotrophs, but yeah. The organism that make their food by themselves, like plants, they do photosynthesis, they prepare their food by themselves. These are autotrophs or chemoautotrophs. Like chemically, like with the help of light, then photoautotrophy, with the help of chemicals and chemoautotrophs. This is all. Now next we have, if we are studying the mode of nutrition, let's see the reserve material. Like in animals, we have glycogen as a reserve material. We store our food, and when we are in need of energy, urgent energy, the body breaks that glycogen into glucose, and these glucose provides us energy. So you know the concept, we are connecting it to everywhere so that we can revise it, and the concept is clear and clear. So let's see the reserve material. Reserve material are inclusion bodies. Double exclamation marks. This is important, like so much important. And the question, like this is a PYQ of need, that these inclusion bodies are non-membranous. Or most appropriately, the option was membraneless. So these inclusion bodies are membraneless. Reserve material of the bacteria, we can say storage granules also. These are also known as storage granules. So this was all about monera, the basic characteristics. Okay, so now we are going to study archaebacteria in detail. So let's see what NCERT says. Archaebacteria have 16 S RNA, and the other usual eubacteria have 70 S RNA. This is Swedberg unit, and this is RNA, DNA, proteins. We're going to study that further chapters, right? Okay. Archaebacteria. These are also known as ancient bacteria. Ancient bacteria. So we're gonna see what makes them so extremely survival and what makes them survive in extreme situations, basically. So these ancient bacteria have cell wall of, I already told you, right? Cell wall made up of glycoproteins. And pseudomurine. This is important. Now there is the plasma membrane. After cell wall, the plasma membrane comes into play, right? It is made up of phytanols. And this is also way too important. Okay, so if you guys remember, then in just some minutes ago, I told you that let's keep it a secret that what makes these archaebacteria so survival, so extremely strong and hard, we can say. So this is the plasma membrane. Plasma membrane, these phytanols, it is highly branched. Highly branched plasma membrane made up of phytanols. This plasma membrane is responsible for survival in extremities, we can say. There we have what the reason is for survival of these archaebacteria. Okay, so as I have told you, like these archaebacteria are most survival. So let's see what examples do NCERT have of archaebacteria. I already told you in the previous few minutes, but let's write it down over here and understand in detail. So Archaebacteria. First we consider thermoacidophils. Thermoacidophils. Thermo. What does thermo means? Thermo means temperature, height. In high temperature, they can survive in high temperature, plus acido means acidic medium. Highly acidic medium also. They can survive in high acidic medium as well as high temperatures. So these can tolerate high temperatures. The example for this is Taq polymerase. You're going to study biotechnology in class 12, so don't worry about that, learning the examples. These are not going to come in exam, but for a basic knowledge you should know, and this is interesting also. Taq polymerase or vent polymerase, right? Next one is halophiles, or we can say halophiles. Halophiles. What do we mean by halophiles? Okay, if you know chemistry, then halo means halogen, and halogens are salt. Salt making, halogen, salt generating. So halo means salt, and halophiles mean they can tolerate extreme salty conditions. Salt ज़्यादा होगा वहाँ पे भी ये रह सकते हैं. Halophiles. Now next and last we have methanogens. Methane is like produced in rumen of cattle, like CH4 to natural gas, or we can say biogas, CH4. They are present in marshy areas. We all know what marshy areas are, so no big deal. They are present in marshy areas, or we can say the rumen of cattle where CH4 is produced. So this was all about archaebacteria. Okay, so now we're going to study eubacteria in detail. So eubacteria have 70S RNA, unlike archaebacteria which had 16S RNA, right? So eubacteria. U means true. U means true. So these are true bacteria, and now the cell wall again is made up of peptidoglycan. And without wasting any time, let's jump into the examples of eubacteria. Eubacteria can be photosynthetic autotrophs, chemosynthetic autotrophs, or heterotrophs. So these are all like bacteria. There are bacteria, once these are present everywhere. So photosynthetic, chemosynthetic, heterotrophs. So let's see it in detail. Okay, so we're going to study photosynthetic autotrophs first. Photosynthetic autotrophs, the example is cyanobacteria. So, ma'am, why first example and not the explanation? Because we're going to study on the basis of cyanobacteria. So, first example, cyanobacteria. Okay, so cyanobacteria. The pigment present in cyanobacteria is chlorophyll A, which is universal pigment, along with the carotenoid B, beta carotenoid. and chlorophyll A are like universal things. They are present every in all the plants and algae, plant kingdom. So next thing, they can be colonial. They can form colonies. Colonial, filamentous. They can form filaments, or they can be unicellular also. Right? They can form colonies, unicellular, filamentous. The next point is they are present in marine water, fresh water, and along with it they are terrestrial. So the cyanobacteria have pigment chlorophyll A. They are colonial, filamentous, unicellular. They are marine, freshwater, terrestrial. So first of all, before going further, let's see, do we know what cyanobacteria is? The other name for cyanobacteria is BGA. What does BGA means from? BGA means blue green algae group. Blue green algae. So they are algae. The bacteria. What are these? Let's go and search for it. I'll tell you this in next lecture. So let's see further. We have studied pigments, colonial, the forms and what. These colonies, if they are colonial, these colonies are surrounded by a gelatinous sheet. If you have studied NCERT, you have saw the diagram of nitrogen fixing heterocysts in that, you must know the gelatinous sheet is all surrounded. Gelatinous sheet. Some fix nitrogen. In the structures called, what I said right now, heterocysts, right? And the example for these are noster, etc. It is according to NCERT. It is. So this was all about autotrophic, photosynthetic autotrophs and cyanobacteria, I can say. Okay, so the next one is chemosynthetic autotrophs. That is the production of food through chemicals. Synthesis of food through chemicals. So let's see what chemosynthetic autotrophs are. They convert inorganic substances like NH3, nitrate, nitrite, etc. I am talking about nitrogen products because this is most important, and these are going to come in further chapters also. So this is important that we know it right now. These convert inorganic substances like these into organic and use the energy in the form of ATP release. We can say that these recycle these nutrients. Recycle nutrients in environment. Nutrients like, of course, nitrogen we already studied, phosphorus, sulfur, iron, etc. So let's know more about it, right? We all know about nitrogen cycle occurring in our environment. How does this work? First we have like ammonia. Ammonia is present over here. This is a reversible process and it forms nitrite first. This is 2. Nitrite, and this is carried out by nitrosomonas and nitrococcus. After that, this nitrate is further converted into nitrate. Nitrite is further converted into nitrate by nitrobacter. This is ammonia. Nitrite. And this whole process is nitrogen fixation done by chemosynthetic organisms, bacteria. This is most important in this chapter. Not chapter, but in this unit. This is most important. Ammonia converted to nitrite by nitrosomonas. These bacteria are important. That's what bacteria is used for, what conversion. So from ammonia to nitrite, nitrosomonas and nitrococcus are important. And from nitrite to nitrate, nitrobacteria is important, that is used for nitrogen fixation. Okay, this is all about chemosynthetic bacteria. Now we are going to study the last part of this topic of this kingdom, monera, and the heterotrophic autotrophs. Okay, so are you ready? This was the easiest part, I guess, but important, most important part. Okay, so this is all about heterotrophs. Not all about heterotrophs, but these heterotrophs are most abundant. This is all theory, so I just skipped the writing part of it. So heterotrophs, most abundant bacteria, uses production of curd from milk, like lactobacillus. So have you seen in your houses like this, like the person preparing food or preparing things in your stuff, like eatable things in your stuff, eatable stuff in your house. When making curd from milk, put some amount of curd in the raw milk and let it sit for all night. Let it sit for whole night. So what does that thing for, and why do these people do that? So the small amount of curd added to raw milk lets the whole curd converted to milk. How do they do that? So the small amount, like the small amount of curd added to the milk is known as inoculum. And this inoculum contains large amount of lactobacillus. And these lactobacillus, when added to the milk and set for whole night, they multiply, or we can say divide. In biology, multiplication and division means the same. So they divide and increase their number and cause the whole milk to get converted to curd. So this was all about production of curd. This was not a chef's class, so this was all. Now next is production of antibiotics. So we all know what was the first antibiotic prepared. That was penicillin actually. And the next is causes bacterial disease. These heterotrophs, eubacteria. causes bacterial diseases. So what these bacterial diseases are: typhoid, cholera, tetanus, citrus cancer. We just have to remember these names, that these are bacterial diseases. In the further chapters, like in class 12, we're going to study all the diseases caused by bacteria. We're going to have to learn it. Now you're lucky that do not have to learn it like by heart. Just you should know that these are bacterial diseases. The bacteria cause diseases. We all know it. So the next part is reproduction in bacteria. The major form of reproduction or major type of reproduction the bacteria uses is fission. Binary or multiple depends, but they use fission. Majority like binary, I think. And some also uses sexual reproduction. Okay, before sexual reproduction, let's know what do they do in unfavorable conditions. Unfavorable condition. When unfavorable condition applies, they form spores, and these spores are highly resistive. Resistive to what? Resistive to environmental factor, the resistance that they should provide, so that these unfavorable conditions do not matter anymore. So these spores are highly resistive. So in unfavorable conditions, they reproduce by spores. These spores flow through water currents. Sorry, air currents. And this causes reproduction. And now we have sexual reproduction in bacteria. So it is the most interesting part. We can say these divide sexually to sex pili. They make a structure known as sex pili. Let's see what sex pili is. Basically, let's feel what sex pili is. So this is a bacteria. This is a bacterial cell. This is another bacterial cell. It has an incipient nucleus and it has a plasmid. Let's make these nucleus, because they do not have a cell wall. I have made the DNA in this way. This has a plasmid. Plasmid has a let's see plasmid as a genetic material for now. It is a vector to transfer the DNA into this part. So this plasmid, it is circular. Let's know it now. This plasmid gets transferred to this another bacteria through sex pili. So this is what we call sex pili. This is the structure known as sex pili. The process is known as conjugation. This process of sexual reproduction that occurs in bacteria is known as conjugation. So now that we're at the end of this lecture, we are going to study mycoplasma. So what are mycoplasma? Already told you, the smallest cells. Membraneless. Sorry, they do not have a cell wall. Lax cell wall. They are pathogenic. Resistant to antibiotics. How? Because these antibiotics act on cell wall, which we are going to study in biotechnology again. So these are resistant to antibiotics because they do not have a cell wall, of course. And the most important point is that these are anaerobic. These are anaerobic that survive without oxygen, in absence of oxygen. Okay, so this was all about monerans, and now we're going to study it further, what protists are, and then virus, and then that's it. Fungi also. Okay, so thank you and have a nice day ahead.